Genetics is the study of heredity and inheritance of characters. Human genetics explores genes, chromosomes, and DNA, which are responsible for different traits and diseases of human beings. Mendel was the father of genetics, and through the study of molecular biology, we have understood that DNA carries all the genetic information in the form of genes that tells us or creates for us a blueprint of life. Genes are located on chromosomes, and the genetic makeup of a human being is called its genotype. But when these genes are translated into proteins and then those proteins have specific manifestations, what observable characters you see are called the phenotypes. Genetics helps us diagnose disorders, understand evolution, and improve medicine. Let's look at the structure of DNA. DNA, or deoxyribonucleic acid, is made up of nucleotides. One nucleotide has a phosphate group, a sugar molecule, and a nitrogen base attached to it. These nitrogen bases can be adenine, thymine, cytosine, or guanine. DNA is a double helix structure where sugar and phosphate make the backbones of these ladders, and the steps of ladders are made by the bonding between the nitrogen bases, where A always bonds with T and G always bonds with C. These nitrogen bases are bonded together through hydrogen bonds, and sugar and phosphate molecules are bonded by covalent bonds. DNA stores all the genetic information, and it guides the synthesis of protein and therefore is responsible for the resulting traits in a human being. DNA replication means creating duplicate copies of DNA so that every time when two cells divide, they have exactly same copies of DNA in them. Gregor Mendel, who is also known as the father of genetics, established the law of segregation and law of independent assortment, in which he established that two alleles or two alternate forms of a gene are separated at the time of gamete formation. Also, one is called dominant and the other is called recessive. So one trait ends up covering or masking the effect of the other. The one that has the visible effect is called the dominant allele, and the one that gets masked is called the recessive allele. And also he said, on a chromosome, if there are different genes, these different genes are randomly and independently going to enter the gametes. So one allele you can get from mother, one allele you can get from father, and they are not really linked. So he established that alleles can exist in homozygous conditions where both the alleles on both the chromosomes are either dominant or both are either recessive, or it can be a heterozygous situation in which one allele is dominant and one is recessive. So there are several traits in human beings that follow Mendel's law. For example, widow's peak, which is the hairline that has kind of V shape in the front of on the forehead, attached earlobe, or the capability of rolling tongue. They follow simple Mendelian rules. In human beings, cystic fibrosis and Huntington disease have been established as one related to the recessive allele, which means this disease will express only when both the alleles are in homozygous recessive form for this disease. And Huntington disease is a dominant disease, which means if you have even a single allele, here you have both alleles for Huntington disease, but if here you have one allele as a in the dominant form, you will still get the disease even if you have one recessive allele, because the gene for Huntington disease is the dominant gene. But Mendelian inheritance is much simpler and have predictable outcomes. Whereas in human population, we see that many traits are much more complex and they don't necessarily follow Mendelian rules. For example, Mendel was clearly able to say that green color is dominant over yellow color in a pea plant. So whenever you make a cross between green and yellow plant, or the plant that has green and yellow pods, the presence of one dominant gene is able to mask the effect of green color, and you will only see green color in the resulting offspring. But then there are situations of incomplete dominance, where if one parent has um, curly hair and the other parent has straight hair, the child can end up having wavy hair. So it's neither of the two extreme conditions, but somewhere in between the two. Then we have already studied that blood group inheritance is a case of codominance and a situation of multiple alleles. So you have three different kinds of allele. One is this, second is this, and third is this. And you can have a combination of any of these three. Plus, we have seen that these two are codominant, which means if IA and IB are ever present together, it would not lead to A or B type blood, but it would lead to an AB type blood. So that is a situation of codominance. Then we have also seen that one particular trait, for example, here the skin color is not controlled by only one gene but multiple genes. For example, here we see that there are three different genes with two different alleles, in total 6 alleles, which are quantitatively going to determine the color of someone's skin. So more number of dominant alleles you have, irrespective of which gene. It can be a combination of these three, or 3 in one and 3 recessive in the other condition. So any combination of dominant genes you get, you will be able to determine how dark someone's skin would be. For example, this chart very nicely explains that if a man and a woman with this kind of phenotype end up mating, there is a possibility of 64 different combinations, and you can see that the number of dominant alleles indicated by the solid black dot, you have more dark is the skin. So here you have all 6 dominant allele, this is the darkest, and here you have all 6 hollow white or recessive alleles, therefore the skin color is the lightest. In human beings, there are some characters that are inherited through sex chromosomes, such as color blindness and hemophilia. So let's look at how that inheritance takes place. So here we have a female, and you know this is a female because it has 2 X chromosomes, and we have a male partner that has 1 X and Y chromosome, that's why it's a male. Now we are looking for the inheritance of color blindness, which is a recessive trait. So this is the color blindness trait. Now when we have any dominant gene that is masking the effect of color blindness, the female will be a non-colorblind or a normal person. In the male, we don't have any small n, so that's why the man is not colorblind. Now there is a separation of gametes, and the male and female gametes are going to fertilize. And let's look at the end result. Here we do not have any color blindness allele, so this female is not colorblind and is not a carrier. This female has 1 normal allele and 1 color blindness allele. So this female is a carrier, but herself she is not going to be colorblind. This male does not have the color blindness allele because color blindness allele is a recessive allele, and this man is also going to be normal. But this man who has only one option, and this allele was actually received from the mother. So although father was normal and a non-carrier, but mother was also normal but a carrier, she ended up giving this single allele for which there is no other option which can dominate or mask its effect. Therefore, this male will end up being colorblind. So although till now we have studied that there are 23 chromosomes that a person receives from his father and 23 chromosomes that he receives from his mother, but along with mother's chromosome, he also receives some of mother's mitochondrial DNA, and that mitochondrial DNA also has certain genes on them. So the contribution of some characters is additional or additionally coming from mother. Now let's say that father has all good mitochondrial genes, but mother has some problem with her mitochondrial gene. So because we only receive mitochondria from mother, the mother will end up passing those problematic gene to all her children. But let's say father has problem with his mitochondrial genes, but mother does not have a problem with her mitochondrial gene, and father does not pass on his mitochondrial genes to the children. Therefore, there is no mitochondrial gene problem in the children if only the father has problematic mitochondrial genes. Human beings have 46 chromosomes, which are in the pair of 23, which you can see here in chart. Out of each pair of homologous chromosome, one has been inherited from father and one has been inherited from mother. Out of these 23 pairs, 22 are autosomes and one chromosome pair, which is the 23rd chromosome pair, is the sex chromosomes. So this chart that you're seeing that gives you the organized profile of individual chromosome is called the karyotype. Chromosomes are arranged as per size, banding pattern, and the position of centromere, and that's how these pairs were made. This chart is helpful in detecting abnormality in chromosome numbers. For example, if this 21st chromosomes had 3 copies, then that situation is called trisomy of 21 and it leads to Down syndrome. Mutations are changes in DNA or changes in the genetic material, which can be in the sequence of DNA or in the chromosome itself. So a point mutation is, let's say, if you have a sequence of base pairs like A, T, G, C, 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 and only one of the bases changes into something else, that will be a point mutation. Now let's say this base pair gets removed or a new base pair gets added, and that ends up changing the whole frame of how you read these triplets of codons, then that will be an insertion or deletion. On chromosome level also you can have mutations. For example, here in the figure you can see a piece of chromosome gets deleted, or a piece of chromosome gets duplicated, or it get inverted, or it get inserted on a different chromosome, or there is a swap. A piece goes from here to the other chromosome and from one chromosome to other. That will be a translocation. Mutations are very harmful, or they can also be beneficial. They can be silent, means they have no effect, but they can also have some effect. Genetic disorders arise when mutations disrupt the normal function of a gene. For example, cystic fibrosis, which is a situation in which some mucus is built around some organs, maybe in respiratory system or in digestive system, and this mutation is a recessive mutation. That means it only expresses when the allele is in homozygous recessive form, because there are some dominant forms of this allele which can mask the effect of this gene, and the person will be a carrier but will not necessarily be affected. Sickle cell anemia is a mutation that causes problem with the hemoglobin of red blood cells. Huntington disease is a dominant mutation. That means even if it's present in a heterozygous situation, Huntington disease will present its effect in a carrier as well. Down syndrome trisomy or Down syndrome syndrome is a situation where you have three chromosomes instead of two chromosomes for number 21, and people end up with Down syndrome, which is a developmental disorder. Detecting these genetic disorders have become much easier with the advancements in the field of molecular biology, where we have now the human genome sequence. We are able to perform all these genetic tests and karyotypes. Evolution in species are the changes that accumulate over generations through the process of natural selection and genetic variations, and that kind of help those species to adapt better to their environment. Human evolution studies how modern human beings evolved from their um, ancestors or from earlier primates. We have evidences from fossils, we have evidences from molecular biology based on the changes in DNA and genes, and we have evidences from the comparison of anatomy of different animals that tells us how these small changes helped human beings to adapt better to their environment. For example, the capability of walking on 2 feet, having a large brain, and having an opposing thumb that helped human beings to grab tools and write and do fine motor skills, and their ability to develop a language. All these have helped human beings to be more advanced than their ancestors. From the discovery of fossils, we have learned that human beings have evolved through different stages, where human beings have passed through the stage of being Australopithecus, Homo habilis, Homo erectus, and finally we became Homo sapiens that we are today. And interestingly, the word sapiens means wise. So we are considered to be the most wise species that exist on this planet. Fossil records provide us evidence of how a species has evolved over time. So if you look at these different relatives of horses, we see that they have common structural features, and slowly some changes evolved that created today's evolved horse. Then we can look at their anatomy. First example is homologous structures. So homologous structures are those structures which look similar but they have different functions. For example, here you will see all these structures have a forearm and then a upper arm, and they have a hand-like structure. But what are their functions? Their functions are different. In human beings, it helps in writing. In dogs, it helps in walking. In birds, it helps in flying. And in whale, it helps in swimming. Analogous organs are those that have different structures but they perform similar functions. For example, look at the wings of all these animals. But in birds, it is actually a limb, the forelimb. In bat also it is a limb, and in butterfly they don't have any bones in them but they are just cuticles. The third example are vestigial structures, the structures which were useful or helpful in our ancestors, but over the course of evolution the higher organisms lose them. For example, this nictitating membrane, which could produce this protective covering on top of eye. So some birds are able to pull this nictitating membrane over their eyeball so that the eyeball doesn't dry when they are flying high and they don't have to close their eyes. But we do not use this nictitating membrane because we can just blink or close our eyes when we are tired or our eyes are dry. And that's why this nictitating membrane is the pinkish structure that you see at the corner of your eye, but it is not functional in us. But this structure is still there. That means In our ancestors, maybe it was useful, but we only have a rudimentary form of it. Otherwise, there is no point of this structure being there in our eye. Why would we even have it if we never needed it? When we look at the embryo of different animals, we see that early embryonic stages are very similar. They are almost, in this picture, you can see that they are almost indistinguishable. Even if it's an embryo of a fish or a chick or a calf or a human being, they almost start with similar looking morphology or structure. But as they evolve or as they go through the stages of embryogenesis, they then start looking different. Even molecular biology tells us that our genes are so similar with chimpanzee that 98% of our DNA is similar to that of chimpanzee. It's only that 2% DNA that makes the difference and makes us who we are. To understand what natural selection is, let's start with an example. So here it's a lighter surface and you can see a moth present here. Now imagine that a bird comes and wants to prey on a moth. The bird would be easily able to spot this moth. But after industrial revolution, when there was so much smoke coming out of industries and all the walls and surfaces and even tree barks would start getting darker, what became visible is this white moth, which you can almost not see here. So when a bird would come, it would be able to detect this moth and eat it up, which is why being darker in color gave the dark moths an advantage of survival. They would never get killed or almost never get killed by their predator, and their survival became more likely. So, natural selection is characterized by all those variations that already exist in the population. So something happens that make that particular trait or character an advantage for the organism's survival. Those advantageous traits are then passed from generation to generation, and possibly over a period of 100 years, as a industrial revolution kept happening, we only end up with black moth, and the white moth were all wiped out because they became so visible over the dark surface. So after 100 years you would think that there are only dark color moths in on this planet, but the fact is there were light color moths as well, but because they became so detectable through the period of industrial revolution, that they became extinct, or they were removed, or they were not selected in the process of evolution. So natural selection of an organism is that the organisms that have traits which are better suited for their environment and help in their survival, those organisms get selected because they had better chances of survival, others get wiped away. And when they procreate, they keep passing on the same characters. So for example, here we see that a giraffe had an advantage of eating apple or fruits from the tall trees. So this long neck gave it an advantage or accessibility to better food resources. And whenever they procreate, they are going to produce the offsprings with long neck. So over time, these beneficial characters become more common in the population and lead to an adaptive change in that species. Now, the natural selection can be of different types depending on of all the variations that existed in a population, what is being selected. For example, let's say all this variation in this fur color of mice already existed. If the conditions or the changes in the environment over only favor darker colors and the lighter get wiped out, then it's a directional selection. If the change happens in such a way that either the darkest or the lightest survive, then middle ones get wiped out, it's a diversifying selection. And if only the one that is in the middle, the intermediate character, has better chances of survival and the extremes are getting wiped out, then it's a stabilizing selection. So, modern human beings, the Homo sapiens, have originated in Africa about 200,000 years ago. They migrated from Africa all over the world, and that is why we have the theory out of Africa. That means all human beings came out of Africa. They adapted to different environments, which is why people have different skin colors, people have different dietary preferences, different tolerances to different kinds of foods, but all human beings share the same DNA. About 99.9% DNA is same in different ethnicities. Other than the chromosomal DNA, the DNA of mitochondria and the DNA on the Y chromosome has really helped us determine our origin and where we came from, what was our ancestry. African populations show the highest genetic diversity, and this confirms that actually all the human beings on this globe came from African lineage. So this knowledge of genetics and evolution help us in so many different ways. For example, genetic testing has helped us determine possibility of disorders or diseases in human beings. We can predict that they might get some kind of cancer or some kind of genetic disorder um, at the time of childbirth, at the time of pregnancy, or even in adult people, knowing if you carry certain kind of genes which make you more likely to cancer help you take some preventative measures. Then because we know what genes are causing problem, we should be able to develop gene therapies to cure those genetic disorders. Pharmacogenomics helped in creating personalized medicines, because if our genes are different and we are producing different kinds of proteins, then not the same drug is not going to work in all human beings, because the drugs are going to target proteins, and if the proteins are different, then that drug might work in one person and not in the other person. So creating more personalized medicine is pharmacogenomics. Then genetic engineering helps in um, changing some genes or modifying some genes, has till now helped us with agriculture and medicine. Although human cloning or has been a bit controversial, but gene therapy in human beings has very high promising results. CRISPR-Cas9 is a gene editing technology that helps us in removing and introducing some genes in the genome of human beings and other organisms. It has really helped people with genetic disorders. DNA fingerprinting has helped in determining paternity, has helped in forensics by helping find the criminals based on their DNA samples. And all this knowledge of genetics and evolution helps us track our ancestry and genealogy. So, to summarize, today we learned that human genetics is the study that covers inheritance, variation, and genetic disorders. We recapped the structure and function of DNA, and we understood that how Mendelian genetics, although it's the foundation of genetics, is not very applicable when we talk about higher organism, more complex organisms like human beings. And we studied how mutations can cause different kinds of genetic disorders. We learned about human evolution and what are the evidences based on which we learn about how human beings evolved. The process of natural selection is a driving force in evolution, and human beings who have evolved over a long period of time, over about 200,000 years, have a common ancestry which probably comes from Africa. And all this knowledge of genetics and evolution helps us in surviving better, in making our lives better by advancements in the field of medicine, biotechnology, and forensics.